Thank you very much. At first, I would like to thank the organizers for inviting me. It's a pleasure to be here. I will speak about the sweet death caused by antibiotics, and I will focus on Clostridium difficile toxins. And in my talk, at first, I would like to give an introduction to Clostridium difficile toxins, and then I will present recent data on factors, intracellular host factors, which are essential for the activity of the toxins. So here is Clostridium difficile. Clostridium difficile is a gram-positive anaerobe spore-forming bacterium. And the problem is really that this is a spore-forming bacterium. And here you can see the spores, because it's really difficult to remove the spores in the clinics, for example. And Clostridium difficile is a cause of CDI, Clostridium difficile infection. And this disease comes in different flavors. We have antibiotic-associated diarrhea, the pseudomembranous colitis, and the recurrent colitis. And the recurrent colitis in 20% of cases is a problem because the patients are treated, successfully treated, and then after 2 weeks or 1 month, the disease will come back. And then it's more difficult to treat. Here, usually the disease starts with antibiotics, and then we see the problems of antibiotic resistance, because the antibiotics are used quite too often. And it starts with the therapy. We have the alteration of the microbiome, and when Clostridium difficile spores are around, we have the colonization, and then the development and the release of toxins. And the toxins cause inflammation and damage of the gut, and the result diarrhea and pseudomembranous colitis. And here you can see an early state that are necrotic areas that are volcano or mushroom-like early areas. This is also shown here. This is a disposition of fibrin and dead cells, and these form the pseudomembranes. And that is a late stage here of the disease. Okay, that is a normal gut, and after infection you have this gut. And actually in the States, 2011, 500,000 cases, and 30,000 died of it. So the people, it's a big problem in clinics, in the Western countries, in the Western world, in the developed world, I would say. We have 3 toxins, and they are essential. Actually, 2 are essential. The major toxin, Clostridium difficile toxin A and B, and we have a minor toxin. However, this minor toxin is found in the hypervirulent strain. These 2 toxins, I will mainly focus on these toxins. These are glucosyltransferases, and they are big toxins, 300 kilodalton. One is called enterotoxin because, for historical reasons, in the former animal model, only this was enterotoxin, but not this toxin. It was cytotoxin, 100 to 1,000 times more cytotoxic. But now it is clear this is most likely the most important toxin, toxin B. And that is the 3rd toxin in the hypervirulent strain. It's different, completely different. It's an ADP ribosyltransferase. Okay, this is the action, a model of the action of the toxins. At first, again, to the structure. We have 4 domains, a glucosyltransferase domain at the N-terminal part, then a protease domain, a translocation part, and a binding part. And this is toxin B, also called TCDB. The toxin binds to receptors, and it's then endocytosed, and we have endosomes which have a low pH, pH of 6, 5.5 even, and then the toxin changes its conformation and can insert into the membrane and translocates into the cytosol. And here in the cytosol, we have inositol hexakisphosphate. This activates the protease, and that is also shown here in on this slide. We have here inositol hexakisphosphate. In the cell, we have 100 micromolar of this molecule, and you can see increasing concentration of inositol hexakisphosphate activates the protease, the inbuilt protease, and that is the crystal structure of the protease. And here is inositol hexakisphosphate, and by an allosteric mechanism, this protease, these are the catalytic amino acids, are activated, and then we split here this part, and that is the biologically active part, and that is released, and then this part can modify Rho proteins that are small GTP binding proteins, and these proteins are inactivated by the attachment of glucose. And here again, the major mechanism, we have the release of the glucosyltransferase, the modification of Rho proteins, and then we have a change of the morphology of cells, because I will this explain in more detail in a second. The cytoskeleton is redistributed from here, from control to the intoxicated part, and in the gut, the result is seen here. Okay, the attachment of glucose is the reason why we have this title, The Sweet Death. I would like to explain a little bit Rho proteins. They are small GTP binding proteins, they are switch proteins, and they are inactive in the GDP bound form and active in the GTP bound form, and in this form they interact with effectors and they regulate the actin cytoskeleton, but not only. So many metabolic pathways in the cells are regulated by Rho proteins. They are similar to Ras protein. Rho means Ras homologous proteins. And important for infection is that the epithelial barrier function is regulated by Rho. Migration, phagocytosis, cytokine production, immune cell signaling, superoxide anion production are regulated by Rho proteins. And the glucosylation had the following effect. The glucosylated Rho protein is no longer able to interact with effectors, and all this regulation is blocked. And some years ago, Feng Shao showed that the modification of Rho protein is even a signal, that is a sensor for activation of the inflammasome, a specific inflammasome, the pyrine inflammasome. So we have some function, loss of function, and also gain of function by the modification of Rho proteins. And here you can see almost the total toxin, the crystal structure, and we have crystallized in collaboration with Georg Schulz in Freiburg. This is the glucosyltransferase in 2006 and 2008, and we had to wait more than 10 years to get this structure by Borden-Lacey's lab. And this is again here in red, the glucosyltransferase. In blue, that is a protease, the inbuilt protease, and this part, this yellow part, is this part, and is involved in the translocation of the toxin into the cytosol, from the vesicle into the cytosol. And actually, it's not known how this can function here. This complicated structure is involved. And this here is involved in binding, but not only this. The binding might be also here. Okay, the question is how such a big toxin, such a big protein can translocate here into the cytosol. And I can give no good answer to this. Most likely, it's a pore formation. That we have shown some years ago that there is a pore formation by these toxins. And the idea is that at low pH, there's a pore formation, and then part of the toxin can go through this pore. And then we have the activation of the protease by inositol hexakisphosphate, and we must have the refolding of this single chain which goes to the pore, and we have the rebuilding, the refolding of the complete toxin. And recently, a doctoral student in my laboratory was able to find factors which are essential, most likely, for this refolding. And these are the TRIC proteins, or Cct proteins. TRIC means TCP1 ring complex, and Cct means chaperonin containing TCP1. That is the same name, the same abbreviation. And the structure is given here, but more precisely here. So this is a real big ball complex, and we have two boxes, an upper box and here a second box. And this box, the upper box, consists of 8 proteins which are dissimilar. They are not identical, they are different. And this box you can open and can be closed independent of ATP. So it's a chaperone, it's a so-called chaperonin, which is here functional in interaction with the toxin. And here are parts, domains of one molecule. We have an upper part, and this upper part is involved in closing the box. And this part here, the equatorial part, is this part. Yeah? And this is a really big protein involved in the binding and refolding of polypeptides. And for example, actin and tubulin depend on these TRIC Cct complex. They cannot fold by themselves. Therefore, actin, you cannot express actin in E. coli, for example, because they have not the chaperonin, and therefore it's not functional. Okay, and here is a simple model how this works. We have here the unfolded protein, and it might be an unfolded domain. This is a second domain, and this binds here at first nucleotide dependent in the presence of ATP to the upper part of this ball, and then we have the closure, and we have the refolding then of the protein in this box, and then we have the hydrolysis, and the box will open again, and the protein is released in the folded form. Okay, and now we studied whether this is also important, this complex, for the toxin. And this is a simple experiment. We partly inactivated the toxin, the glucosyl transferase domain, only one domain, at a higher temperature for 10 minutes. Then we cooled down it in the presence of the chaperonin and ATP. And after some time, we tested the activity, the glucosyl transferase activity. And the result is shown here. You can see that in the presence of CCT or TRIC, there is much more activity because we have the refolding. Other chaperones, such HSP90 or only BSA, are not able for the refolding as given here. So a major effect, this refolding effect. And this is ATP dependent as shown here. In the presence of ADP alone, it's not possible. You need ATP, which is then hydrolyzed. Okay, there's one inhibitor of CCT trick. And the advantage is that this inhibitor can enter cells. And we now tested, is there an effect in intact cells? The data before were in vitro. And we intoxicated the cells. And here you can see the control. Here the cells, it's not easy to see in the, here in the background. But here you can see the rounding up after the toxin. The rounding up, typical the destruction of the actin cytoskeleton. Here the inhibitor has no effect on the control conditions. But here the intoxication is inhibited, is blocked by this inhibitor of CCT, indicating that CCT is essential for the intoxication, which is also shown here. And then, of course, today, one inhibitor is not enough to show the effectiveness of this system. We were able to knock down CCT5, a part, essential part of this complex. And here you can see the same. That are the control conditions. This is the toxin TCDB rounding up of cells. And then we knock down CCT5, and you can see there is no intoxication. Another type of toxin, ADP ribosylating toxin, is still toxic here. There's rounding up of cells, indicating we have a specific effect. Now, that is again the model, and the idea is that CCT acts here, but the precise point is so far not known, but it is essential. And we have a lot of glucosylating toxins, more than 10, and there are most likely much more. And we studied a lot, and we found that almost all these toxins depend on TRIC and CCT. And this means that it's a general mechanism for glucosylating toxins. And that might be important also for the development of new treatment against the toxins. Okay, this is a summary. Bacterial glucosyltransferase toxins like Clostridium difficile toxin B interact with TRIC CCT in vitro. TRIC protects the toxin against heat, inactivation in nucleotide-dependent manner. Inhibition of TRIC by the inhibitor blocks the cytotoxicity, and knockdown of CCT5 protects cells from glucosylating toxin. So TRIC CCT is essential for the action of glucosylating toxins. So this was done on the 3rd floor of this building, and most important for the studies was Markus Steinemann. And he was his supervisor, Thomas Young. We have nice collaboration, especially for this study. Andreas Schlosser was very important. We have, of course, nice collaborations in Freiburg and also in other places. And I thank you for your attention.